Hey guys, Miss Riggs here. I wanted to do things a little bit different this week with our vocabulary. I really wanted to give you guys a foundation to actually understand the vocabulary a little bit better than just copying down the definitions. So what I'm going to do is go through the definitions and kind of elaborate on those words so that you completely, hopefully, understand those words. As you go, remember, I want you to copy down the definitions as well as write a sentence with each one of your vocab words, and I'll check those off in class tomorrow. So let's go ahead and get started. Okay, so our first vocab word is bacteriophage. And bacteriophage is a virus that takes over a bacterium's genetic machinery and directs it to make more viruses. So what does that mean? So basically what we know of a virus is it is not actually a living thing because it needs a host in order to reproduce. So this process really helped us kind of understand how genetics worked and how DNA actually works. So what we found was these bacteriophage, and you can see them here on this bacteria. And what we know about bacteria is it is a single-celled organism and it is microscopic. So we cannot see it with our naked eye, right? Because it is a prokaryote. So if you see this, what looks like a very large bacteria, but as we know, it's not. We see even smaller viruses attaching to it. And so these viruses are attaching to it in order to use it as a host and to reproduce these viruses. So what we found out by understanding the different elements that are in a virus is that we didn't really understand if protein or DNA was the basic function of genetics. And so we tested the elements within DNA as opposed to the elements within the proteins. And what we found was what was injected was the DNA. So this was really the building block of the study of DNA. And this happened in the 1950s. So it's a very interesting study. But bacteriophage are those viruses that do take over this large bacteria here. So our second vocab word is nucleotide. And nucleotide is the smallest unit or monomers that make up DNA. Here we have a picture of a nucleotide. And the only difference on these nucleotides is going to be the bases here. And we'll talk more about that as we go through DNA and talk about the unit. But these bases will be different based on what kind of nucleotide we have. So these nucleotides come together and they form a rung of the DNA. So the smallest units that make up DNA is a nucleotide. The next vocab word we have on our list is a double helix. And this is two strands of DNA that wind around each other like a twisted ladder. And this just basically describes the physical characteristics of a DNA. So you can see it's kind of like a twisted ladder or a double helix here. Our next word is DNA polymerase. So polymerase is going to be an enzyme, basically. This is a group of enzymes that bond the new nucleotides together. And from what we've learned about enzymes, we should know that these enzymes are basically a catalyst that cause a function to occur. So a group of enzymes that bond new nucleotides together. So basically they're going to build the DNA molecule with these nucleotides, and this is DNA polymerase. Base pairing rules. The base pairing of the two DNA strands always pair up in the same way. So we talked about, when we talked about nucleotides, we stated that the base is going to be different. And so these bases are going to pair up with other bases. So there are 4 different bases when we're talking about DNA. So this is adenine, thymine, cytosine, and guanine. So adenine and thymine are always going to pair together. So they're like puzzle pieces that kind of form together to form a base pair. Cytosine and guanine are the other two that are going to come together. So as you can see, this always occurs this way. You're never going to have an adenine with a guanine or say a cytosine with a thymine. They're always going to pair together. So that's the base pairing rule. Replication, the process by which DNA is copied during the cell cycle. So we learned in our last unit when we learned about mitosis and meiosis that this happens in interphase, right? So these DNA molecules are going to be copied in order to eventually copy a whole cell. So that's DNA replication. So RNA, which is ribonucleic acid, as opposed to DNA, deoxyribonucleic acid, and I'll explain the difference in the name in just a minute, but this acts as a link between DNA in the nucleus and protein synthesis in the cytoplasm. So you can think of RNA as kind of a temporary copy of DNA. It's basically going to be used and then it's going to be destroyed. So basically, there are a couple of differences between RNA and DNA. So RNA has an extra oxygen group on it, so hence the name ribonucleic acid instead of deoxyribonucleic acid. But it also changes one of its base pairs. So instead of having adenine, guanine, cytosine, and thymine, RNA is going to have adenine, guanine, cytosine, and uracil. So we're replacing thymine with uracil in our bases. And we'll definitely explain more of that as we get into our unit. I just wanted to give you a broad concept of RNA as opposed to DNA. So our next vocab word is transcription. And this is the process of copying a sequence of DNA to produce a complementary strand of RNA. So we are transcribing the DNA into a strand of RNA. And again, we'll talk more about this as we get into our unit, but that is transcription. RNA polymerase is similar to DNA polymerase. It is an enzyme that bonds nucleotides together in a chain to make a new RNA molecule. So the same thing as the DNA polymerase, it is basically putting the puzzle pieces together in order to create a whole strand of RNA. So next we have messenger RNA, and messenger RNA does what it sounds like it does. This is also known as mRNA, and this is an intermediate message that is translated to form a protein. So basically, it's a message that it's going to take on to form another protein. And then we have ribosomal RNA or rRNA, and this forms part of ribosomes. This is a cell's protein factory, as we learned in our unit on cells, and we learned about different organelles, and ribosomes make protein. So ribosomal RNA is going to make protein. And then transfer RNA, or tRNA, this is going to bring amino acids from the cytoplasm to a ribosome to help make the growing protein. So again, as we learned before when we talked about biomolecules, amino acid is going to be a monomer of protein. So that's what we are making proteins out of it. So transfer RNA is going to bring those amino acids and create a larger protein. Okay, so that is all our vocab words for this unit. So make sure that you understand. If you have to pause and go back or start playing this again in order to understand the words, please do that. Go ahead and make sure that you make your sentences as well as writing the definitions because you will have a vocab quiz. Thank you for watching the video, and I will see you guys in class.